Hey everyone, hope you are all doing very nice and good. So, today we are going to discuss about embryology of an eye. So, let's move on. So, before moving to the embryology of an eye, we should know what is human eye. It is an organ that reacts to light, allows vision, provides 3D image, give color vision, and perceive intensity of light. Either it's dim or light. So, it is an organ that provides us vision. Anything we see is due to the human eye, right? So, we will see that how this eye is developed or how the development of the eye starts. There are basically three stages for the development of the eye. One is embryogenesis, organogenesis, and differentiation, right? So, what is embryogenesis? It is a stage in which the primary organs are developed. Or you can say that the base of every eye structure is developed in the embryogenesis, right? The complete structure isn't developed right now, only the base or the rudimentary organs are just developed, or the base of organ is developed, that this structure is cornea. It's not completely developed, but it is cornea, right? And this structure is lens. Yeah, this structure will made lens, but this is not a complete lens right now, right? So, or embryogenesis is the stage in which only the base of the structures is made. Then the organogenesis is the stage in which a complete organ develops, right? If we have discussed in embryogenesis that the base of every organ or every structure is made, then that base will continue to develop into further layers. For example, I have said that in the embryogenesis, cornea was just the base of cornea was developed, or we have just marked that yes, cornea is visible, cornea is developed right now, right? So only the base of cornea is developed, or we can say that right now only that development of cornea has started. But in organogenesis, what happens that the complete structure develop, right? All the layers of the cornea develops, lens is completely developed. So in organogenesis, a complete structure develops, right? And in differentiation, what happens that all the structures are made in organogenesis. Now we can differentiate whether it's lens, whether it's cornea, whether it's optic nerve, optic head, nerve head, it's retina, vitreous humor, and all the structures are visible, and they are well differentiated. So these are the three stages of the embryogenesis. Let's move on. So let's move on to the development of eye, that what is the development and how the development starts. So over here you can see a flow chart that what happens basically. Fertilization is done. After the fertilization, a cell is formed, right? This cell is known as the ovum. Okay. So first what happens? Fertilization starts. This fertilization when is done, it forms a cell which is known as ovum. Then what happens? This cell starts to multiply. Any cell that starts to multiply, it will form a blastula. Over here you can see it was just a single cell that it started multiplying, and it is forming a blastula. And this layer is the margin or the membrane of the blastula is blastoderm. So what happens that blastula passes through three gastrulation process. This gastrulation process forms three germ layers, right? What happens basically? Fertilization. After fertilization, ovum cell multiplies and forms blastula. That blastula passes through the gastrulation process and forms three germ layers. These germ layers are ectoderm, mesoderm, and endoderm. Now, what these layers help to form? The ectoderm helps to form eye and major nervous systems, right? Central and peripheral nervous systems. So ectoderms helps to in the development of the eye and the nervous system. Mesoderm helps in the development of gastrointestinal tract and other structures, and endoderms helps in the development of the epithelium of all the structures, right? So epithelium is made from the endoderm, and mesoderm is helped in GIT tract, and the ectoderm is basically helping in the development of eye. So because we are discussing about embryology or the development of eye, so we'll just discuss ectoderm over here, that how ectoderm changes into the eye. So before, we have already discussed that ectoderm is a basic layer that helps in the development of the eye. So ectoderm further has some layers, right? So there are three layers of the ectoderm, which is surface ectoderm, neural ectoderm that is connected to the brain or the forebrain and all, and mesenchyme, which is between surface and neural ectoderm. We'll discuss it over here in this diagram. Before moving to the diagram, I'll just tell you that it starts from the fourth week of gestation, right? Or the 22nd day of gestation. At fourth week, or after the 20 after fourth week, or at the 22nd second day of gestation, what happens over here, that the first step is, before going to the first step, we'll just discuss that what were the layers of the ectoderm. Over here you can see this blue line or blue membrane. This is the neural ectoderm or the forebrain, which I have already discussed. The ectoderm which is connected to the brain, that is the neuroectoderm or neural ectoderm. And these black lines, these are the membrane. This is the surface ectoderm which is on the surface, right? And between surface ectoderm and neuroectoderm, we have the mesenchyme. So this is between the neural and surface ectoderm. Now what happens at 22nd, after at fourth week or after the 22nd day of gestation period, what happens that at the neuroectoderm, is the formation of optic sulcus or the grooves. What are optic circles? They are the hollow pouches, right? Hollow spaced pouches that will form and connect to, over here you can see there is a connection between surface ectoderm and neural ectoderm with by or via optic sulcus. So there will be hollow patches on the both side of the junction between neuroectoderm and surface ectoderm. So these pouches are known as the optic sulcus. So first step is that there will be the optic grooves or sulcus formation. Now what happens next? After these grooves are made, what will happen to these grooves? That they will start invaginate or elongate. You can see over here that over here there were only pouches, but now what happens, they are started elongating, right? They start elongating or they are invaginating, you can say, or elongate and form optic vesicles, right? So they are moving in the both sides. And now you can see over here in the next picture that these optic grooves are turned into a vesicle, more big pouches. So these are now the optic vesicles. So what were the first step? In first step, there is a formation of optic grooves. In the second step, there is a formation of optic vesicles. Now moving on to the third step, we'll see there will be the connection of the optic vesicles with the surface ectoderm. Over here, you can see that these vesicles move or elongate to the surface ectoderm, and they'll touch or they will be connected to the surface ectoderm in the both sides. You can see now what happens, that they will elongate to the surface ectoderm. And now what happens basically, when they will be connected to the surface ectoderm, they will make two structures, right? Because we are saying that in embryogenesis, this is a first stage, you can say, that in embryogenesis the base of all the structures starts to make, right? So what is happening over here, that this surface ectoderm and optic vesicle connection will make lens placard or the lens base and optic cup, and it will start making optic cup. Now how they will make lens vesicle and lens placard or optic cup, we'll discuss in fourth step. But over here what happens, there will be the elongation, further elongation, and it will connect to the, or it will touch to the surface ectoderm, and they'll stick to it, right? So now in the fourth step, what happens, you can see over here, there is an invagination. There is a first step after being connected to the surface ectoderm. There will be the invagination of surface ectoderm in the optic vesicle. Over here you can see it's moving inward direction. So this optic vesicle, sorry, will maintain the shape which is becoming over here, right? So what happens when it invaginates, optic vesicle also invaginates. In the second step, you can see in both direction it is happening. Invagination is happening in both of the sides, because we have two eyes in our body. So over in our face, body, or human being have two eyes, so that is why it is happening both sides. So invagination will occur, it will invaginate to the point, then a circular structure will be formed over here. So you can see that this is the optic cup. A cup structure is formed from with the help of or from the optic vesicle. A optic cup is formed, and inside it there is a structure, but there is a layer of surface ectoderm that is moving in. A point will come when it will be cutted off, right? And the surface ectoderm will connect to its proper, maintain its structure. So what happens, this circular thing is the lens placard. This surrounding this red circle, or which is surrounding, is the cup form, so which is optic cup. And this surface ectoderm will be converted to the cornea. So what happens over here, you can see it is a cornea. It is a cornea which is a surface ectoderm which has taken its place again. The circular portion over here, which is cut, is the lens placard. Or now, after the it has been separated from the surface ectoderm, it will be now lens pit, and the thing that is surrounding it, the structure which is surrounding it, is the optic cup. So it will protect, this optic cup will protect the lens pit, right? So what happens next? This type of structure is formed after this all phenomena, right? So what happens, you can see this is the neuroectoderm. Mesenchyme is in between the optic cup and the lens placard or lens pit. This is a primary structure of the cornea, and all these structures are made, right? So there is this line, you can see it is a choroidal fissure, or you can say that basically what happens, that lens is a vascular structure, we all know that, but during the period of gestation, there is lens is the vascular structure which provide nutrients to the lens and the structure surrounding it, which is hyaloid artery. Okay, so hyaloid artery passes from the brain, comes to the lens, and it provide nourishment to the lens, but after some time it starts to disappear, right? So after it is disappeared, that portion which is now empty and there is no hyaloid artery, that becomes a choroidal fissure, that space. So if I'll show you over here, this is a optic cup and there is further lens pit. I have shown you over here like that. So what happens over here? This hollow space from where the highlight appeared, and this space now is the choroidal fissure. So, okay, the choroidal fissure. Now, this outer layer is of optic cup is the outer layer of optic cup, and this inner one is the inner layer of the optic cup, and this whole is the optic cup view, so that you can understand choroidal fissure very clearly, right? So now what happens after the this all happens till this part, right, where cornea, base of the cornea, you can just say that epithelium and Bowman's layer of the cornea is formed right now, right? And lens pit is formed, optic cup is formed, choroidal fissure. And one more thing, over here you can see that there is elongation of the optic cup, right? Over here, if I'll show you that this structure is now elongated, and it is forming a tube-like structure, which is the optic stalk, right? So this will be connected directly to the brain or the forebrain. So now we'll move on. This all happens till the from fourth week to the sixth week, right? After the 6th week, now what happens after the 6th week? So over here, at 7th week of gestation, what happens, that optic cup formation is done, right? Optic cup is formed. So now what happens, that there will be the maturation of RPE, or retinal pigmented epithelium, which is formed from the outer layer of the, you can see over here, from this outer layer of the optic cup. So maturation of retinal pigmented epithelium will be from this outer layer of optic cup, right? And then there will be the development of neurosensory retina, which are the rest of the 9 layers of the retina, right? So that will be developed, that will start to develop, and it will be from this inner layer, right? So this inner layer will be turned into the neurosensory retina, and the outer layer of optica will be the retinal pigmented epithelium, right? So now, so now only the optic cup formation have turned to the retinal layer development, and corneal base like epithelium and Bowman's layer have been made, and lens pit is developed. So rest of the structures are remaining, right? So now there will be the formation of other ocular structures, right? So what, how they'll form, they will be formed by the multiplication of neural crest cells and mesenchyme, so that other structures can form. More probably, the first structure that will be made from this multiplication will be stroma and endothelium of the cornea, because epithelium and Bowman layer is already made from the surface ectoderm, right? So after the multiplication of neural crest cells and mesenchyme, what will happen, there will the formation of the stroma and an endothelium of the cornea, so that cornea, all layer of the cornea are fully developed, right? Now, what is neural crest layer or neural crest cells? Over here, you can see that we will move backward. So moving backward over here, you can see this is the neuroectoderm, right? So the cells on this forebrain or neural ectoderm are the first layer, or the cells present on the top or the surface of the neuroectoderm are the neural crest cells, and here is the mesenchyme, right? So this mesenchyme is between all the structures. So now there will be the development of other ocular structures from these neural crest cells and the mesenchyme present, right? Now, at what happens at 8th week of gestation? Now, other structures are in development, are made, right? Now, rest of the structures are remaining. What the most important structure is, that how you are going to connect that eye to the brain. So there will be the development of optic nerve, which will be due to the ganglion cell differentiation, or the multiplication or the differentiation of the ganglion cells. So ganglion cells will differentiate or multiply, and they'll start the development of the optic nerve, right? So there, at the 8th week of gestation, there will be the development of the optic nerve, and then optic chiasm will be formed. Now what happens over here, when we know that in real pathway we have discussed that after optic nerve we get optic chiasma. So over here, when optic nerve ganglion cell, after the differentiation of the ganglion cell, optic nerve has been formed, there will be the formation of optic chiasma, so that it can connect to the brain. So what happens over here, that the axons of the optic nerve, when the axons of the optic nerve contacts to the brain, there will be the formation, there will be the formation of optic chiasm will be started over there. Then the third thing that will start will be the appearance of the Muller cells, right? So Muller cells will start to appear, and they'll move, their radial fiber will move in different direction, they'll move inward. When the Muller cells will move inward, or when the radial fiber of the Muller cell will move inward, they will form internal limiting membrane, or we can say that is the layer of the retina. So that layer of the retina will be formed by the Muller cell, and when the Muller cell radial fibers of Muller cell will move outward, they will make the external limiting membrane. So there are 10 layers of the retina. One is retinal pigmented epithelium, and rest of the 9 are the neurosensory retina, which is in the neurosensory retina. So two limiting membranes are formed with the help of Muller cells, and rest of the neurosensory retina is made from the inner layer of the optic cup. So now all the development is done, and how all the structures are formed, or from which structures all the ocular structures are made. So cornea, the epithelium and Bowman layer of the cornea is developed from the surface ectoderm. The stroma and endothelium of the cornea is made from the multiplication of crust cells and the mesenchyme. Sclera is made from the multiplication of neural crest cells and mesenchyme. As I have already told you that the rest of the structures were made by the multiplication of these structures. Choroid is made from the inner vascular layer of mesenchyme. So mesenchyme have a vascular and avascular layer. So choroid is the layer that absorbs the uh, that absorbs light and has a vascular supply, right? It contains vessels. So that is why this is formed from the inner vascular layer that have blood vessels of the mesenchyme that surround the area of the optic cup. So over here, yes, over here you can see the area which is surrounded by the optic cup, the area that optic cup surrounds, right? So over here, these structures will make these mesenchymes, inner vascular layer of mesenchyme, and the neural crest cells, right over here, they will start to multiply to form the choroid. Ciliary body, we know that ciliary body have epithelium, so both layers of, if it's either it's pars plicata or it's pars plana, so all both the structures epithelium is made from the anterior part of the optic cup. Optic cup's front portion will make ciliary body epitheliums, and the stroma, blood vessels and ciliary muscles which helps in accommodation, they will be made from the vascular layer of the mesenchyme that will have blood supply and or the blood vessels. And the iris epithelium is made from the marginal cells of the neuroectoderm, and muscles that either dilator and sphincter are made from the anterior part of the neuroectoderm. The front part of the neuroectoderm make the muscles for the iris, and the blood vessels and stroma makes the vascular layer of mesenchyme, right? And vitreous, we have two type of vitreous. When it's with the eye is not fully completely made or complete, right, the development is not done, we have a primary vitreous, and when it's done we have the secondary vitreous. So the primary vitreous is made from the neural crest cells. So what they are saying is, when the structures aren't made, a gelatin material is formed, that is a primary vitreous. So when all the structures aren't made completely, there is a gelatin material that protects all the structures from any damage or harm. So that gelatin material is the primary vitreous. Whereas the secondary vitreous is made from the inner layer of optic cup and lens vesicle. So that inner portion of the optic cup and lens vesicle will make the secondary vitreous. Retina is made of by, I have already told you, there are two basic layers of retina. One is retinal pigmented epithelium, and the other one is neurosensory retina that have further 9 layers. So the outer layer of optic cup makes retinal pigmented epithelium, and inner layer of optic cup makes a neurosensory retina. And the eyelid is by the reduplication of the surface ectoderm above and below, so of the cornea. The cornea was basically epithelium, and Bowman was made from the surface ectoderm, but that further surface ectoderm will reduplicate and make a more surface or more new structure, that is eyelid, which will protect the eye, protect the cornea and the eye from any harm. So that is the eyelid. So this was all about embryology of an eye in a very easy words, and hope you all get it very clearly. If you have any query, you can ask in comment section, and thank you so much.